Let us begin the session on surveillance for infectious diseases with the topic of current status and issues of surveillance of infectious diseases transmitted from wild animals. I am Kimiko Okabe. I serve as the chair. I am from the Center for Biodiversity, Forestry and Forest Products Research Institute of Forest Research and Management Organization. Our first speaker is Dr. Ken Maeda from Department of Veterinary Science, National Institute of Infectious Diseases. Dr. Maeda. Dr. Okabe, thank you very much for the introduction. I am Ken Maeda from the Department of Veterinary Science of National Institute of Infectious Diseases. This is a topic on current status and issues of surveillance of infectious diseases transmitted from wild animals. And the theme given to me is on expansion of tick-borne infectious diseases by wild animals. Severe fever with thrombocytopenia syndrome, SFTS, is the topic that I'm going to cover mainly. And this is a borrowed material. This is the classification of ticks and tick-borne infectious diseases. There are many different species of ticks, and what we focused in our talk is Exodidae. There are tick-borne infectious diseases found in Japan that include Japanese spotted fever, Q fever, Lyme disease, and borreliosis, as well as tularemia. They are bacterial infections, whereas tick-borne encephalitis and Cassanu forest disease are classified as viral infections. When we talk about tick-borne infectious diseases, bacterial infections have long been well known in Japan, but recently viral infections have become better known. The fact that there's no established therapy and there's not a clear understanding about the route of transmission lead to the focus of attention. It's not that tick-borne infectious diseases are new, but rather people have come to know better about viral infections. What this shows is the data from 2013 to 2014. This the incidence of tick-borne infectious diseases, including Tsutsugamushi disease. This is the incidence of different prefectures in Japan. SFTS, that I'm going to talk mainly, is found mostly in western part of Japan, and Japanese spotted fever or rickettsia also is found mostly in western part of Japan, while Lyme disease and tick-borne encephalitis are found in Hokkaido, and Lyme disease also is found mostly on high mountains. This is very interesting because depending on which tick-borne the particular disease is different. Some tick live in northern part of Japan, some ticks live in the western part of Japan. So among the tick-borne infectious diseases, they can be classified in terms of types of ticks that borne the disease and the types of animals that the tick prefer to feed on. There's a correlation among these factors. Among various tick-borne infectious diseases, SFTS is prevalent in western part of Japan. Also, you have Japanese spotted fever in western part of Japan, but SFTS prevalence peaks in April-May, where Japanese spotted fever has a peak in autumn. So depending on the developmental stages of the tick for in transmission, the outbreak timing is different because there are different stages of development among the tick from larva, nymph, and adult. That leads to the difference in terms of the peak prevalence. Therefore, the types of tip, the developmental stage, the preferred animal for blood feeding, all these factors need to be considered comprehensively when we think about tick-borne infectious diseases. This is the SFTS infection cycle. All stages of tick carry this SFTS virus. And these ticks feed on wild animal. The important thing is, why is it that we focus on wild animal? Of course, cats and dogs are bitten by ticks, so is the human, but not many tick bites they suffer from. Maybe stray dogs and stray cats may have many tick bites, but not as much as in case of wild animal. Just because you are bitten by the dog, you get diseased, but that would not transmit the virus to other ticks. But wild animals are bitten by many ticks. So wild animals serve as the starting point for many ticks to get antibody prevalence. So the virus is borne by the tick, but wild animals serve as the starting point or origin to make many ticks positive for antibody. In case of SFTS, humans get infected, but pets like dogs and cats get infected. A cat has a very high fatality of as high as 60%. So this is a dangerous disease for cats and of course to humans. Furthermore, the owner of the pet and veterinarians can also get infected. One of the characteristics of SFTS is that it forms a very complex ecosystem among ticks and all wild animals, in this case mostly with mammals. As I mentioned earlier, the tick carry the virus, and this is the photo of female Amblyomma testudinarium laying eggs. As you can see, there are thousands of eggs. If this female is infected and carrying a virus, then thousands and tens of thousands of eggs become positive, and they grow into larva, nymph, and adult. So the disease spread through this tick cycle. And another important topic in this talk is, as I mentioned earlier, this is the photo of a deer hunted, and there are so many ticks on the body surface of a single deer. Several hundred ticks are feeding on the animal. If this deer is SFTS virus positive, then several hundred ticks, by feeding on the blood, will also carry the virus. So this is a tick-borne infection, but the infection is transmitted from the wild animal to a large number of ticks. This is another characteristic of the disease. And as a result, humans get bitten. These are the photos of tick bite. You have a tick on the surface, you have erythema, you have papilla, and you also have blisters. So humans are bitten by the virus carrying a tick and get infected and develop a disease. The important thing is that many of the wild animals don't develop a disease. On the other hand, humans develop a severe disease. That is what we already know. The important point here is that this is a data from Dr. Anzai of Oita Prefecture. On the left is the data from Dr. Anzai's patients, the patients who had tick bite, and this is the number of patients of SFTS nationwide. They have good match between the two. Oita is a warm place, so it starts early, but you have almost none in February, but you see tick bites in March starting and increasing in summer. Similarly, we have SFTS patients more in summer. When we consider SFTS as a human disease, we can clearly see that there's an increase in the number of cases when ticks become active. Now the question is, which ticks bite? This is important. Which ticks would bite humans? Ticks have preferences in terms of animals they feed on. And this is Amblyomma testudinarium that account for the majority. Also, there's a report from China that Hemaphysalis longicornis carries the virus, and we also have Hemaphysalis flava. In short, Amblyomma testudinarium prefer humans. And this tick also prefers wild boar, whereas Hemaphysalis longicornis prefer deer and cows on relative terms. Therefore, the preference of the tick on the animals they feed on is greatly linked to this infectious disease. Now let's look at the role of wild animals in SFTS infection, which is the topic of this lecture. This is the study of deer in Yamaguchi Prefecture. As you can see, the antibody prevalence among the deer in Yamaguchi is gradually increasing. We started collecting data from 2010. The important point is that during the last about 5 years, almost all deer have become antibody positive. This is a noteworthy fact, or it could be understandable because many ticks bite wild animals. I cannot imagine that throughout a year, quite a large number of ticks would bite a single deer. That means if there's a positive individual, then most of the animals could become antibody positive. This is where you still have low antibody prevalence, but then later entire deer, almost all of them become positive. This is based on different weight. The newborn has higher antibody prevalence. This is because from the maternal side, but as they become larger in size, they become antibody positive in larger percentage. So in Yamaguchi since 2015, the antibody prevalence has almost plateaued. So after a certain time, all deer become positive. With the increase of positivity, then in around 2012, for the first time in Japan, a case was reported. This was a human infection. So between wild animal and ticks, virus was intensified as there's an exchange of virus back and forth. And then we see a human incidence. That is clearly shown here. After 2012, every year, almost every year, we see incidence among human patients, and we see an increase recently. This is preliminary data, but this is nationwide. In the north, there was almost no deer with antibody prevalence, whereas in the west, where there are many patients, more than 10% of the deer are antibody positive. So when you have wild animal with high antibody prevalence, we see human cases. If you look at the prefectures with no patient, you have low antibody prevalence, but if you have cases develop among humans, you have high prevalence rate among the deer. I mentioned that antibody prevalence is increasing in Yamaguchi. This is Kanto region where there is no SFTS patient, but the data last year shows an increase of antibody prevalence. So most likely, as we study wild animals and we see the disease spreading among wild animals, we can presume that in the near future we will see a human case also in Kanto region. This is the data of the study on animals other than deer, and this comes from Tanabe City of Wakayama Prefecture. Most animals are positive, as you can see here. In case of monkey, which is close to humans, 17% are infected. And we have many samples of raccoon, so let's look at more details. In raccoon, the antibody positivity has jumped since 2014. And just like in Yamaguchi Prefecture I referred to, this is where in 2015 we see a human case reported. That's when we see an increase in the antibody prevalence among raccoons. If you look at this in more detail, the raccoons of older than 2 years, 80% of them are positive. That means almost all raccoons are positive. When they were born, they are not infected, but after a year or two, most of them become positive. When they were young, just like in case of deer, they had antibodies from the mother. And as you can see, since April 2015 in Wakayama, almost all the raccoon of the age over 2 are positive. This is Wakayama Prefecture of different regions. Raccoons from the mountains have high positivity, but less in towns and near the shore. So SFTS outbreak is seen in the mountains between wild animals and ticks. That's what we can clearly learn from the data of raccoon. The important point is that this is a rather complicated story, but when you have virus in the blood of wild animal, a tick would feed on that blood and get the virus. So we look at the amount of virus in the blood in the raccoon serum. In April to May, when there's an outbreak, 3.4% of virus detection was observed. This is rather a high number. This is among healthy raccoons. And of course, these raccoons may be bitten by ticks, and all the ticks that bite these raccoons would receive virus, and the disease or the virus would spread. So far I talked about wild animals, and now let me talk about pets, particularly cats. As you know, in urban setting, cats are kept indoors, but there are many cats that are kept loose. So there are often cats suffering from SFTS. What I need to say is that at last we have a SFTS case in cat in Shizuoka. This is the borderline of human incidence, but this year in Shizuoka we had a cat SFTS. That means a cat or a wild animal was inflicted. So we can say that there's already a heightened risk in Shizuoka Prefecture. The data that I earlier showed you about the deer was in Kanto, so most likely we can presume that there will be a human incidence in this part of Japan. Last year we had more cat SFTS than human SFTS. This year we didn't have much human incidence, but we have much more among cats. So wild animals and companion animals see outbreak of SFTS, and by getting that data we will be able to identify human risk. Lastly, since SFTS is seen in Japan, it is in category 4 and BSL is 3. Very similar to SFTS is Crimean Congo hemorrhagic fever, which is in category 1 and BSL level is 4. They are very similar, and these dangerous diseases are very close to us, going back and forth between ticks and wild animals. We really need to keep this in mind. This is not the only infectious disease, but we really need to have to work hard on countermeasures against the tick-borne infectious diseases. What I like to say is that tick-borne infectious diseases are quite dependent on animals, in particular wild animals. So we need to know more about the conditions of wild animal and companion animals, and by doing so we can know the amount of risk of human infections. So the future studies are going to be very important. Thank you very much. Thank you very much, Dr. Maeda, for the presentation. Next, I'd like to invite Dr. Yasuhiro Yano from Department of Medical Zoology, Faculty of Medical Sciences, University of Fukui. Dr. Yano, are you ready? Please begin. Thank you for the introduction. I am Yano from Department of Medical Zoology, Faculty of Medical Sciences, Fukui University. Thank you very much for inviting me to this session. The topic given to me is searching for the timing of infection from the location of pathogens in ticks. As mentioned before, there are various tick-borne pathogens including virus, Rickettsia, Frasizilla tularensis, Borrelia, and protozoan, through which various diseases develop. Our group studied ticks' capability as a vector. To be specific, when these diseases occur, we visit these sites, we collect ticks, and when we identify the species, and to see whether or not the ticks carry the pathogens, to identify the cause of a transmission. In case of emerging infectious diseases borne by ticks, identification of route of transmission is very important. By identifying the route, we hope to contribute to the prevention by providing information as to which part of the infection cycle we need to tackle with. As was mentioned before, we have Japanese spotted fever, SFTS, Lyme's disease, tularemia, tick-borne encephalitis, recurrent fever, anaplasmosis, Babesia microti. There are various tick-borne infections. Mostly they are categorized in Category 4 in Infectious Disease Act. Category 4 disease is not transmittable between humans. Humans are infected through animals and food. Physician who diagnose the disease must immediately report to the nearby healthcare center. It's important to identify the number of patients. Today I'm going to talk about Japanese spotted fever, Lyme disease, and Babesia microti. These are the ones that we have some experiences with. After the discovery of SFTS since 2013, we have many inquiries about tick bites, and as was mentioned before, amblyomma testudinarium cases are increasing, and we have Haemophthalitis longicornis and Haemophthalitis flava, and these species have high probability of being vectors. This is the female of amblyomma testudinarium that prefers humans. This is after blood feeding. You can see by the size of my hand how much it enlarges in size. In Mie Prefecture, we accompanied a hunter, and when you see how wild boars are hunted, as you can see, there are many amblyomma testudinarium on the surface of the wild animal. And these wild animals come close to human habitat, and these ticks are released, and they lay several thousands of eggs that creep up the grass. And this is a case in which numerous bites by amblyomma testudinarium larva was identified. We are involved in this. Our student after graduation found this, and this was the report prepared. You can see there are numerous larvae of amblyomma testudinarium. So the wild animal brings these ticks and cause the disease to develop. And we identify pathogens from ticks using various methods. This is hemolymph test. This is mostly for rickettsia. It is like the blood test of a tick. Or we can grind and inoculate the ground tick into culture cell or medium to isolate various viruses. And nowadays, PCR is a mainstay, and with PCR, we detect the genes. In these two tests, we need to grind and emulsify the tick. So we cannot tell in which tissue or organ the pathogens exist and whether or not the pathogens are active. Therefore, we use electron microscope to detect pathogens in the body of the tick. And I'd like to report to you on these results. This is a hemolymph test. You cut the tip of the leg. The body fluid is placed on a plate, stained, and seen. This is rickettsia. This is the tick, and we focus on salivary gland as it feeds on the blood, and this is the salivary gland. You can see dark blue in granules, and then there's light blue granules, and there is granulous portion. This is the morphology. And then we code wild rat, and on the ear you can see that ticks are attached. Under the electron microscope, you can see that the tick is penetrating deep into dermis to feed. Earlier there were darker blue and light blue granules. These granules are released out of the salivary gland that serve as a cement-like substance to be attached on the animal. And this is the hypostome of the mouth of the tick. This is how deep it penetrates into the skin. This is the sensory hair to find where to bite. Now let me first talk about Japanese spotted fever rickettsiosis. This is about the disease and the current status. The first report was made by Dr. Mahara of Annan City in 1984 on 3 cases who were well-Felix OX2 positive with erythema and fever. Well-Felix reaction is used for rickettsiosis, particularly Tsutsugamushi disease. It is a test method. OX2 positive indicate spotted fever. So this was unusual. And later, he continued to research and propose the new term Japanese spotted fever. Later, Dr. Uchida from Tokushima University called this pathogen Rickettsia japonica. 10 years later, our predecessor, Dr. Takada, identified and isolated this strain for the first time from Dermacentor taiwanensis. So far, from 4 genera and 9 species of ticks, more than 300 strains were identified. This is the data I received from NIID. Reporting started in 1999 as a category for infectious disease. In 2019, there were 318 cases with 13 deaths. Altogether, there are 3,108 cases with 44 deaths. During the last few years, every year, we have more than 300 cases reported. This is rickettsia isolated from the Japanese ticks. We have Al japonica, Al helupatica, Tumari, and Halongiangenesis. These are the ticks with the case reports. There are some others, but we do not know the pathogenicity of them. Rickettsia japonica was isolated from Haemaphysalis longicornis, Haemaphysalis hysteresis, and Haemaphysalis flava. They have preference to humans for blood feeding, therefore their likelihood of being a vector is high. Now let's see Haemaphysalis hysteresis and Dermacenta taiwanensis. We are going to see inside their bodies under electron microscope. These are male and female of Dermacenta taiwanensis. These are male and female of Haemaphysalis hysteresis. These are male and female of Haemaphysalis longicornis. This is rickettsia isolated from a patient inside a culture cell. You can see very active division inside the cultured cell. One of the features of spotted fever is that the pathogens go inside the nucleus. This is the cross-section of rickettsia. The length is about 2 microns. The diameter is around 0.5 microns. The feature is that it has a cell wall. By confirming the morphology, we now look into the salivary gland of the tick, and we identify the similar organism. This is the cell without granule. This is salivary gland cell with granules. You can see the white circle. This is another feature of the spotted fever rickettsia. There are granules released from the salivary gland, and you can find pathogens around them. The pathogens also are in the nucleus and proliferating. There are some in the myocytes, and also there are some in the central nervous mass or in the brain. There are some in Malpighian tube and also in midgut epithelial cells. And you can find them in ovary. So Rickettsia japonica is found in all the tissues and cells that we observed in the tick. Next, I'd like to talk about Babesia. This is babesiosis. The first human case was reported in 1970 from Nantucket Island of the United States with rat infesting Babesia microti. The vector was Ixodes scapularis. This is the vector of Lyme disease in the United States. In 1999, Dr. Saito from Kobe University identified the first Japanese babesiosis among the anemic patient who had a history of blood transfusion, and the protozoan was found included in the transfused blood. And together with Dr. Saito, we identified Ixodes ovatus using PCR in Hyogo Prefecture. So this is patient blood. It is in the state of so-called malaria. In erythrocytes, you can see infestations. So this is under electron microscope, and this is the erythrocyte. This is the Ixodes ovatus, and in the salivary gland, there are granules. And sometimes you can find swollen cell without granules. The nucleus is enlarged, so it is rather unusual. Under electron microscope, it's murky, it's not very clear. But as we study the literature, we can see that this is the Babesia before differentiation. We use the rabbit to feed, And if you look at the salivary gland, granules are almost gone after release, but you can see swollen cells and there are spots. Under electron microscope, you can see the pathogen. These are also pathogens. There are many that you can find which have already been differentiated. With the blood feeding stimulation, this pathogen promotes its development. Now let me talk about Lyme borreliosis. Simply put, this was a report of the cluster in Lyme region of Connecticut in 1977. Arthritis cluster was reported, and Bergedorfer identified the unknown spirochetes. This is a spiral pathogen, and this reacted with the serum. And Johnson called this newly Borrelia burgdorferi. Kawabata and Baba in Japan for the first time reported a case with Erythema chronicum migrans in Myoko. When it is tick-borne, the spirochetes would spread over the skin, spreading the erythema. And Baranton identified Garini and Afzeli, which also are found in Japan. And we know that Ixodes parsalcanthus serve as the vector. And in Japan, Kawabata isolated B. japonica specific to Japan from the Ixodes ovotis. And in Japan, we had 19 cases reported and 13 cases reported in 2018. The number is small, but in the United States, there are 20,000, 30,000 case reports. This is the European strain under electron microscope. It is in the spiral form. Its feature is that there is a flagellum. By identifying this form, and if you look at the tick in salivary gland, we couldn't find one, but in the lumen of midgut, we identify the pathogen. This is under expanded view. This is not Ixodes ovotis, but this is inside the ear of a rat. This is a peripheral nerve. And inside, this is the expanded view. You have flagellum. Next to Schwann cell, we were able to identify Borrelia. So this could be the evidence that it causes neural damage. And as you go upstream, this leads to central nervous system. So this is a photo that really shows the evidence. So the summary is that in case of rickettsia, it is found in salivary gland even before the blood feeding. And the adult tick has about 7 days for infestation from the bite to detachment. So rickettsia can be infesting and transmitting the disease in early stage. Babesia requires 2-3 days for the growth, so it would transmit the disease in the middle part of this feeding duration. Borrelia was found in the midgut, so if we assume that the pathogen moves on to the gland, then this may happen in the latter part of the blood feeding period. But since they were in the midgut, then if you have a tick bite and if you try to remove the tick and damage the insect, the inside Borrelia could infest and transmit the disease, so you should be very cautious. Then the question is, at what stage and which vector SFTS virus it would be active on. We are working on this, but it's not easy to get positive ticks. So this is the end of my talk. Thank you very much. Dr. Yano, thank you very much for the presentation. Thank you very much. Now, as the third speaker, myself, Kimiko Okabe, serve as the third speaker. I am with the Center of Biodiversity, Forestry and Forest Product Research Institute, Forest Research and Management Organization. Forestry and Forest Product Research Institute carries out comprehensive research on a broad range of topics related to forest, including ecology, forestry, and forest products. In the framework of One Health, we recently initiated a research on the health of ecosystem and of animals, particularly of wild animals. In my talk today, I will speak mostly on the results of our ecosystem management project to control zoonosis that our institute is involved in. I will talk about wild animal and SFTS as an example of tick-borne infectious diseases, about what we learned through the participation of wild animal researchers in the field. I will also talk about ecosystem management for infection control from the perspective of forest researcher and how that could become effective and in which area it could work. Currently, global environmental issues are becoming more and more serious, and these include reduced biodiversity, deterioration of ecosystem, and climate change. In the meantime, zoonosis continues to rise with infection spreading. In our research, we collaborate with National Institute of Environmental Studies, National Institute of Infectious Diseases, Tokyo Metropolitan University, and Yamaguchi University. As Dr. Maeda said, since 2012, tick-borne infectious diseases, SFTS, severe fever with thrombocytopenia syndrome, started to spread in western Japan, and it is now moving to north and east part of Japan with increasing number of affected regions. As was stated earlier, just like Lyme disease, another tick-borne infectious disease, which has become an issue in North America and in Europe, increase in deer population led to the increase in ticks, which in turn led to the spread of this infectious disease. Since the relationship between deer and tick is critical, risk is high in hilly and mountainous areas. Among those infected, those above the age of 60 have high incidence and mortality. The people actively engaged in forestry or those manage wild animals as hunters are the ones that fall into this age group. And they are known to have a very high risk. Together with National Institute of Infectious Diseases, National Institute for Environmental Studies, and Tokyo Metropolitan University, we are carrying out research on specific method as to how we can incorporate wild animals into the measures to control SFTS. First, let's think of the factors that cause the increase of zoonosis that derive from wild animal, including SFTS. For example, in Japan, with social change, there has been a rapid change in the distribution and density of wild animals after the war. For example, there has been a large decline in the number of hunting license holders. On the other hand, there's a huge increase of abandoned farmland that would feed or serve as habitats of wild animals, such as deer and wild boar. The decline in human population in these hilly and mountainous regions means there is less threat to the wild animals in their habitats, and that is considered one reason for these animals to increase their population. Now let's look at the result of analysis on SFTS incidence. The result of spatial analysis on Miyazaki Prefecture's cases carried out by Dr. Yasuo and Dr. Nishimura is shown on the map. The spatial analysis on incidence of the disease and landscape of the area proved from scientific point of view that the risk is high in mountainous area and flatland close to hills. I need to point out that our ecological study excels in a comprehensive methodology of analyzing ecological features, topography, and land use so that we can conduct analysis on landscape. Once we know where the high-risk area is, we can explore ecosystem management or wild animal management specific to the area, so the results give us clear identification of the area to work on. Using Yamaguchi University's data, our research institute, in collaboration with Yamaguchi University, analyzed the impact of deer density and land use on the deer and virus. The result predicted that the higher the deer density, more deer become antibody positive. Namely, there will be an increase in the number of deer infected with the virus. Thus, the viral density also is increasing. On the other hand, where there is more residential land, that means as we come close to the urban area, antibody prevalence of deer declines. To further development of this research, we believe it necessary to consider human density for the perspective of transmission to humans. The analysis clearly and scientifically showed the importance of deer density control when dealing with SFTS virus. As our first speaker, Dr. Maeda said, deer is an important animal in considering SFTS, but a variety of other wild animals are involved. With the collaboration between Nature Center Tanabe City, Wakayama Prefecture, and Yamaguchi University, it is presumed that the exotic species of raccoon is strongly involved in the spread of infection. The joint research at our Forestry and Forest Products Research Institute revealed that the distribution of antibody-prevalent raccoons is expanding at the speed of 4 kilometers a year. Dr. Maeda already talked about the importance of raccoon. Raccoon is a wild animal, but its preferred habitat is close to human habitat, not in the mountains. So, the rise in raccoon's antibody prevalence may suggest the possible transmission of the disease to other animals, such as pets and companion animals, through raccoon. Furthermore, these red dots indicate antibody-prevalent raccoons, and expansion of these dots from 2012 to 2016 suggests the possibility that the raccoon may be spreading the virus towards mountainous area, that the virus may be transmitted from areas close to human habitat to wild animals such as deer. That is the possibility we have been considering. With the collaboration with the pathological research, we are currently carrying out a study on effective wild animal management or comprehensive wild animal management. So far, I talked about the introduction of our ecosystem management approach. In collaboration with Ministry of the Environment and Ministry of Agriculture, Forestry and Fisheries, we at Forestry and Forest Product Research Institute have been working on the research and measures against deer as they cause serious impact on agriculture, forestry, and on biodiversity conservation. Currently, with the additional engagement of the experts from medicine and veterinary medicine, our research focuses on deer management from the perspective of infectious disease control. Our approach of ecosystem management focuses on risk mitigation with cost in mind. The first thing we need to understand for such methodology is, as I already mentioned, we need to identify the factors behind the issue. To establish concrete measures, we need to explore the target of the management. We must predict risk and synergy to develop appropriate management method. I mentioned the importance of deer as the target animal of the management, but we must at the same time promote the tick management as one of the wildlife. So, we studied whether we can manage tick by working on deer as our target animal. As a result, it became clear that there is correlation between deer density and tick density. For example, according to the study in Tochigi Prefecture, the tick density increases with the rise in deer density. However, not all tick species increase, but we saw some increase among genus Hemaphysalis, including H. flava, H. megaspinosa, and H. kitaokai. Also, the increase in deer density changes the makeup of the tick species in the region. This is a result of multivariate analysis on the similarity of tick population of different areas. We learned that the composition of tick species becomes similar as the deer population becomes more dense. As is shown in red or orange circle, the increase in deer strongly contributes to the similarity of the tick population. We also found that there's a great influence from the vegetation that cover the ground. If we just look at the impact of vegetation and compare tick density in the forest and forest edge against forest road, where almost all vegetation is removed, as you can see in the photo in the very right, the tick density of the road is extremely low. What we can say from this result, from the management perspective, is that appropriate vegetation management is effective in controlling the tick density. On the other hand, complete removal of vegetation poses greater problem on biodiversity. Therefore, we are currently studying what would be the most appropriate method to manage vegetation while predicting the trade-off. One of the measures for appropriate management includes the use of natural enemy. Through our research on mice, we happened to discover the pseudoscorpion, a small arthropod of about 5 mm in size, live in the mouse nest, and it attaches on the mouse coat to migrate. We also discovered that tick is pseudoscorpion's favorite feed. Pseudoscorpion feeds on tick in such a way that the tick becomes almost void of body fluid once sucked by the arthropod. However, you cannot just spray or scatter natural enemy in a particular area. This is the very function of ecosystem in the nature. One of our challenges is to maintain forests in such a way that these natural functions are not lost in the process. So far, I have shared with you some of our research results. Recently, various scientific verification studies have been conducted to answer the question of whether ecosystem management is an effective measure for zoonosis control. We now come to know its importance. For example, on a global scale, we now know that the reduction in tropical rainforests greatly increases the risk of zoonosis. On the other hand, the increase in human population density raises the risk of zoonosis. Although changes in climate or wildlife or expansion of farmland are greatly linked to the risk of zoonosis, there is no linear relationship where one increases and the other declines. We learned that the relationship is very complex. Thus, the preservation and protection of ecosystem have been discussed and reported in various occasions recently. The preservation of ecosystem, in other words, means preservation of biodiversity. Now the question is whether the preservation of biodiversity can reduce the risk of zoonosis. This point is still under debate. Hypothesis on dilution effect is proposed to show the mechanism. For pathogens as well as for vectors including ticks, there are desirable and undesirable hosts. The increase in wildlife biodiversity would increase not only desirable hosts but also undesirable hosts, thus reducing the possibility of these pathogens and vectors to reach their preferred hosts. That is the hypothesis of a dilution effect. Hypothesis has been proved for Lyme disease and malaria using mathematical model. But that doesn't seem to mean that it is applicable to other diseases. We also know that the reduction in risk does not directly translate into the reduction of patients. Ecosystem is very complex, and its actual mechanism relates to interspecific interactions, including what the animals feed on, and is extremely difficult to understand. But we now know that the preservation of sound ecosystem as a whole is an important measure to control zoonosis and its risk. We believe we have a lot of work to do to prove the effectiveness of ecosystem management. As far as cost is concerned, we believe the ecosystem management is most cost-effective, because ecosystem management is the method specializing in prevention. The purpose of ecosystem management is diverse, and it includes climate change control, preservation of biodiversity, and reduction of damages on agriculture and forestry. Therefore, we believe we can offset the cost. With all this in mind, we need to collaborate with other research fields and develop appropriate methodology. And this collaboration itself is One Health. Our message is that ecosystem management is supported by science and collaboration among scientists. By promoting transdisciplinary approach involving public administration, managers of ecosystem in the field, as well as medical professionals, we hope to implement suitable ecosystem management and promote zoonosis risk mitigation. Thank you very much. Now, I'd like to invite the last speaker of our session, Dr. Koichi Goka from Ecological Risk Assessment and Control Section, Center for Environmental Biology and Ecosystem Studies, National Institute for Environmental Studies. Dr. Goka, are you ready? Yes, I'm fine. Then, please begin. Thank you. I'd like to begin. Now, I'm from National Institute for Environmental Studies located in Tsukuba City, Ibaraki Prefecture. The institute is under direct supervision of the Ministry of the Environment with a mission to study and design measures for global warming, biodiversity preservation, and pollution, including PM 2.5. I belong to the Biodiversity Preservation Project, and my research team study biodiversity-related risks, including alien species, agricultural chemicals, and wildlife infectious diseases. Today, I'd like to focus on wildlife infections in my talk. With globalization, infection or human infectious disease have become serious issue around the world. Ministry of Health, Labor and Welfare is principally in charge of this, not the Ministry with the Environment, with researchers with medical and pathological background. But recently, we ecologists need to get involved because the issue is not only related to humans but also to wildlife. One example which drew much attention is chytridiomycosis.
that only affect amphibians. It is amphibian emerging infectious disease. Since the 1990s, the infection rapidly spread among amphibians, causing the extinction of many precious amphibians, and it became a serious biodiversity issue. Chytrid fungus infests on amphibian skin to cause the disease. Since 1980s, there was pandemic leading to massive amphibian death deep in jungle. In Japan, the pathogen was found in a foreign pet frog imported from abroad in December 2006. It was a big topic those days, as there was a fear of the pathogen entering Japan and endangering Japanese amphibians. So quickly we initiated the project. We took swab samples from the amphibian to collect DNA to see whether the chytrid DNA is contained in the solution through PCR. This is just the same method of PSA tests used for COVID-19. We studied the distribution of the fungus and analyzed DNA variation. We collected the fungal DNA information from across the world, not only from Japan, to study, and carried out phylogenetic analysis to find the source of the pathogen. To our surprise, we found that the pathogen originated in Japan. The pathogen spread around the world from Japan, and a large number of non-immune animals died in overseas. So that's what our phylogenetic analysis suggested. Later, through follow-up studies by many overseas researchers, led to the conclusion that it is almost certain that the origin of the pathogen is East Asia, including Japan. The vector of the pathogen is American bullfrog, originating in North America, an alien frog. As the frog was delivered around the world to be eaten as food, the pathogen from Japan spread all over the world. It is presumed that ecotourism and field trip brought tourists deep in the jungle, through which the pathogen entered the deep jungle. Nowadays, we see TV programs in which people catch wild amphibians with bare hands, and it is a concern that such act may have a risk of spreading infection among wildlife. The important suggestions we obtained through the chytrid research are that pathogens and parasites have their original home. Chytrid fungus originally lived in Japan and East Asia. In the original habitat, pathogen and parasite have history of coevolution with the host and have symbiotic relationship. When they were brought out or when humans enter the area, the spillover of the pathogen causes infection. The research data of chytrid clearly shows the mechanism of infectious diseases in human society. The control of infectious diseases require not only medical science and pathology, but also biology and ecology. I believe it is necessary to develop new cross-disciplinary study of infectious disease ecology. Tick, fungus, virus, so-called parasites and pathogens are in principle disliked by humans. The only purpose of their study was to eliminate them. But as you can see in the example of the chytrid fungus, these pathogens have long been evolved with other animals on the earth long before the emergence of mankind. Moreover, they played a critical role, that is, if an animal population greatly increases and becomes too dense to tip the ecosystem balance, the pathogen would hold on to the population, cause infectious disease, and reduce the population size, thereby recovering the ecosystem balance and letting the animal evolve with resistance. They serve as natural enemy. Their role is similar to that of a lion that feeds on slow-moving zebra, leaving faster animal to continue to evolve. This means the parasite play an indispensable role as a modulator in the ecosystem, and when we consider biodiversity, we must include the parasite and pathogen comprehensively. As I said, there's a variety of animals, and every animal carries a variety of infesting viruses. As we humans create a wave of development forcing it into their world, it is inevitable that they come into contact with humans, and more spillover of new virus cause serious infectious diseases to human society. So that's the ecological mechanism of the current emerging infections. All the viruses that cause serious problems recently, including SARS, Ebola, hemorrhagic fever, and HIV, used to live among wildlife. When humans entered their arena, the viruses came bursting out into the human society. In fact, the number of so-called emerging infectious diseases are increasing year by year. More than half of them is zoonosis, namely diseases derived from wildlife, and about half of them are by viruses. So emerging infection viruses continue to increase in the human society. And this data from a paper published in Overseas Journal this year shows that it is a complex tree network, and each dot indicate type of animal with different colors. The lines indicate the virus common to particular animals. The size of circle indicate the number of viral species that a particular animal carries. Larger the circle, higher the number of pathogenic virus that animal holds. The connections between circles mean the animals share the viruses. In the center, there's a big cluster of blue circles, which are livestock, meaning domestic animals. And they have the largest animal of infectious viruses. Surrounding the livestock connected with lines are animals that provide viruses to them, which are carnivores, rodents, and bats. What we can learn from these results is that the areas that keep the livestock are encroaching deeply into wild animal habitat, causing viral transmission. As a result, some viruses that evolve in the livestock become new human-type virus, leading to emerging infectious diseases. This is the mechanism that this drawing shows. SARS-CoV-2 is the state-of-the-art zoonotic virus. Analysis on the origin of the virus is underway. According to the most recent data from a paper in Nature, we come to know that the origin is among bats. There's a long history of catching and eating bats in the hinterland of Asia. It is suspected that such act led to the release of virus. As you know from media report, COVID-19 still is a great threat across the world. Particularly in the United States, there has been an enormous increase in prevalence and death. The world is still dominated by COVID-19, with infection continue to expand all over the world. It continues to be disastrous. As of day before yesterday, the total number of cases is 72.2 million, with more than 1.6 million deaths. This may be the first virus in history of mankind that brought pandemic with such a speed. The bad news is that the developed nations in the northern hemisphere is in winter, with a far greater expansion of the cases, much more than during the first wave. It is reported that Japan is in the midst of the third wave. If we relax measures against the diseases, the incidents start to rise. If we don't do anything like in the United States, that incident continue to go up and up, meaning there should be some measures taken. Some mentioned the possible attenuation of the virus, and this was cause of people to become less vigilant. But just a few days ago, there was a shocking report from Kanagawa, a young patient who was very mild, who was under quarantine in hotel, suddenly worsened and died. The group from University of Tokyo has data that shows the possible increase in virulence of the virus. The virus that causes SARS, which is similar to SARS-CoV-2, came from bat and civet and turned to human type. This virus is less active in inhibiting interferon, and so it disappeared without becoming a serious issue. But SARS-CoV-2 is a potent interferon antagonist with a high risk of developing serious disease. In Ecuador, they found a variant with a very high potency against interferon. So it is possible for virus to become more virulent, and we should keep vigilance. We should be watchful for fulminant type, the case I mentioned earlier. Question is, what should we be thinking about now? Some say COVID-19 is just a common cold, but we should not forget the fact that it's only been a year since the discovery of the unknown virus, and there's still a myriad of unknown risks. It has extremely high infectivity and environmental adaptability. Influenza is seasonal with its outbreak in winter, but COVID-19 spread rapidly from countries in the north to south in just over 6 months. Regardless of temperature and humidity, it spread instantly, so it is highly infectious. Also, since there is a wide variation in symptoms from mild to severe, making it difficult to treat. It affects the immune system and infects various organs, and there have been many cases of after effects. So we must consider this disease to be different from the ones we know so far. And since there is no reliable treatment, just a single seriously ill patient can put a huge burden and risk on the medical field, overwhelming the health system. Some say we should live with COVID-19 and resume economic activities. But since the virus causes subclinical infection without preventive measures, the endemic will continue forever, and we will always be fearful of serious outbreak. Some use terms such as with COVID or symbiosis with COVID, but we should realize that it does not mean we can ignore the disease. Since anyone can get infected anytime, we should not only protect ourselves, but we must take altruistic actions not to infect others, or we cannot stop the infection. Our economic activity should be conducted with masks, with social distancing, until medicine prevails over the disease. It was said it's not easy to strike the balance between infection control and economic activity. It is difficult because you try to balance this system. This is the notion belonging to a so-called old normal. We must think of economic development as an extension of infectious control, explore a new mode of economic environment and economic development. The old normal in which people get together to eat and drink is no longer possible. In the new system of economic development, you eat alone, wear a mask as a part of fashion, you change the way you work and have fun. We must think of new ways of economic activities. In the meantime, the virus continued to evolve. It was reported recently that a new variant was found among Danish mink farms, and massive culling of the mink is underway. If we successfully overcome COVID-19, even if that is the case, there will be a continuous supply of new virus from animals. This is the new influenza virus that caused pandemic in 2009 called swine flu. The virus is still under evolution. We know recently that a new H1N1 type is causing massive outbreak among Chinese wineries. If it evolves into humanized type, the virus would become more virulent, causing serious pandemic, and the currently available vaccine may not work, as alerted by researchers. We must be prepared for the continued fight against these viruses. What was revealed through COVID-19 is the fragile international cooperation. It has been a serious issue that WHO, an international body, was slow to declare state of emergency, and there was no effective measures implemented, and global pandemic took place. The weakness of WHO is not new. We have seen a similar situation in 2014 during the pandemic of Ebola hemorrhagic fever in West Africa. Dr. Umar Khan for Sierra Leone provided patients' blood samples to CDC. Using the samples, DNA was analyzed to identify the process of pandemic, and the paper was later published in Nature. Unfortunately, the physician contracted the disease while treating patients and died at the age of 39. The virus spread not only in Africa but also to Europe and the States through international airport. And there was a concern that virus may enter Japan, and that was in 2014. Dr. Umar Khan strongly urged WHO for intensive international quarantine, but WHO did not take immediate action because in 2009, at the time of swine pandemic, at that time, WHO was quick to declare state of emergency, but the disease ended without much disastrous consequences, but there were more economic damages. WHO received a barrage of criticism of overstatement and consequent economic damages. As WHO perceived this as a case of failure, the organization hesitated to declare emergency at the time of Ebola pandemic. So for the current COVID-19, people say the declaration of emergency came late because of economic and political concerns. My topic today is on ecosystem and COVID-19. Let me summarize the reason why emerging infections like COVID-19 is still attacking human society. Ecosystem has a cycle. The plant use solar energy to make nutrients which herbivores feed, which is then fed by carnivores. All dead bodies are decomposed by bacterium and fungus into inorganics, which then become the nutrients for plants. All life is circulated without any waste in the system. The important rule in the ecosystem is that a stronger creature has smaller population size. As you go up the hierarchy, you have smaller share of nutrients. So inevitably, the higher the order of the predator, the smaller the population. So such pyramid makes the system stable, but the emergence of mankind disrupted the system. With a huge biomass of 7.7 billion, the human is at the top of the ecosystem, consuming every possible resources of the biological world, leaving smaller share to those below, leaving the population to decline. This is the deterioration of biodiversity. Since solar energy alone can sustain this system, human dug fossil fuel which they use to supplement energy and material production. This was new to the history of the earth, and the natural world is unable to decompose all the emitted waste. So the waste accumulated and caused population. A huge amount of greenhouse gas and heat energy used to be absorbed by natural forest, but now there's too much to absorb, and the consequence was global warming. Global warming, excess waste, and deteriorating biodiversity, these three major environmental problems all come from humans as a giant biomass of huge consumption and huge waste. They are caused by the enlarged human activity. What happens then is inevitably ecosystem will try to regain the balance. There's an excess population with high density which needs to be reduced in number, so the natural enemy would evolve. Emerging infectious disease viruses come as a natural enemy from the natural world. They enter the human society in huge number as natural enemies because from the viral perspective there's an abundance of feed for them, one of which is the COVID-19 virus. We must recognize COVID-19 as the inevitable rule of nature. In order to get out of such circumstances, what we must think of is that, first and foremost, we must not allow further deterioration of biodiversity, realize symbiosis with nature, otherwise the human society may not be viable anymore. We must not forget that preservation of biodiversity is security of human society. We need to have a full review of biodiversity. Biodiversity was the strongest enemy for human ancestor, which was a weak and naked ape. With wisdom, mankind created community to help and support each other to prevail the fight against wild animals to enjoy the current prosperity. In principle, biodiversity can transform itself into something that can attack humans anytime when humans are not vigilant. Namely, the preservation of biodiversity does not mean you should take good care of living creatures. We should stop glorifying the nature. We must realize all life, including virus, is trying to catch us off guard to attack us. We must understand that the true coexistence with nature can be realized through the relationship of equal terms. The concept of zoning to clearly demarcate human share from wild animals' share is the right way of coexistence with nature. What we need to think toward future is we must move away from the current resource exploitation type of globalization. We must transform the society into decentralized, localized, sustainable society. As a first step, we must establish a lifestyle based on the notion of local production and local consumption. This is another data from a paper published this year in Overseas. The line thickness represents supply chain trade volume loss due to COVID-19. This in turn means there was such a huge supply chain in the past. As you can see globally, the supply chain was promoted centering on China, US, and Europe. This is the ultimate globalization. As we pursued economic benefit and efficiency, the tragedy suffered from that was that most of the medical masks were manufactured in China, and when China's activity was suspended due to COVID-19, there was mask shortage across the world, instantly causing health service disruption. We must realize that the threat from COVID-19 was brought about not by reasons, but rather by the disaster caused by excessive globalization. What is symbolic is the New Zealand with a very thin line, and this country was quick to establish COVID-19 control measures, reducing the new incidence to a single digit now. We must reconsider globalization with the concept of SDGs. We need to promote sound globalization based on decentralization, localization, and autonomous state. Japan should move toward that direction promptly as we are facing crisis. We are faced with a variety of natural, social, and economic threats, not just emerging infectious diseases. We must re-establish independent and solid nation right away. Otherwise, we cannot protect the country, nor can we lead the world. That is what we must think of. Once again, by introducing a variety of technologies, we need to create decentralized society and become strong, independent nation and exercise international leadership for the creation of sound, secure, and safe international society. That is the responsibility and challenge for Japan. I have a YouTube channel on these topics, so please visit and have a look. Lastly, my hobby is to draw ticks and insects using computer graphics. As Dr. Okabe mentioned in her talk, the malicious tick has a natural enemy, which is a chytrid. This is a drawing of them. I likened this chytrid to Amabie and end my talk hoping for successful COVID-19 control. I have a published new book. Please buy one. Thank you very much. Thank you very much, Dr. Goka. Now we'd like to begin the discussion session. In the presentation, with zoonosis as a keyword, we covered such topics as pathology, microorganisms, pathogen, vector, host animal, ecosystem, society including human behavior, and global environment as we broaden the scope. At this point, we'd like to come back to the topic of zoonosis as a keyword. And I'd like to ask each one of you from your perspective of your own specialty and based on your experience. We'd like to ask you about the difficulties and challenges that you have faced so far in control of zoonosis. If there's anything that you're looking for that has not been found, like a new breakthrough. And those of us who are involved in environment, including myself and Dr. Goka, collaboration with zoonosis, it's relatively new. So including this new collaboration, what are the difficulties that you are facing? Can we ask each one of you to comment on this, starting from Dr. Maeda? Well, if we look at this topic from the perspective of veterinary medicine, people say that veterinarians need to take good care of wild animals, but because of the healthcare, we focus on companion animals and we pay less attention to the wild animals. When it comes to tick-borne infectious diseases, there are researchers who have been studying them with established field, but here in Japan, people come to realize its seriousness and danger in terms of the infectious disease. This was the area that people paid less attention to. It's not that it was done on purpose, but it was less focused. But on the other hand, once again, we have come to realize among the researchers and related parties about its importance. As Dr. Goka mentioned, including the outbreak of COVID-19, as researchers, we feel frustrated that we have been letting COVID-19 incidents to continue. It is a new area and there are so many things that we do not know. There are many unknowns. And because of these so many unknowns, the concept of One Health, which is the theme of this forum, I believe is very important. It really is important to have collaborations with experts from all different disciplines, and that's what I've been feeling during the last few years. I hope this is answering your question. What I mean is that we need to have collaborations the type that we have now. Thank you very much. Now that it was mentioned that there was less initiative for tick-borne infectious diseases, Dr. Yano? Yes? When it comes to tick-borne infections in humans, not much was known historically. But for Tsutsugamushi disease and rickettsiosis, for many years our predecessors have supported us in terms of how to promote the research and methodology. But people tend to think that ticks belong to the arena of veterinary medicine, and then came Japanese spotted fever with quite a number of deaths. And it is only during the last 2 decades that we finally come to know of this. It's not that we have more incidents, it means we have more people knowledgeable in this field. And it took us 2 decades. But still some people are missed, they die, sometimes it's too late. So early diagnosis is important. And for rickettsiosis we have a therapy. For viral infectious diseases, it's not that we have good antiviral drugs. For SFTS, we only have about
500 cases, difficult to carry out a clinical study. So that's one issue. How can we protect the human health from infectious diseases? Although they are rare diseases, some are highly fatal. We need education, and we need to get involved in that type of education. That's what I feel. So SFTS, for those of us involved in the tick, we do not know whether tick really is the cause, but it was rather a shock for us to see SFTS. Thank you very much. Those of us involved in this research of biodiversity, the collaboration with zoonosis and the conservation of biodiversity is very new. Not many people in Japan are involved in that initiative. Dr. Maida earlier said that there was a lack of initiatives on working on wild animals. To start with, as we look back after the war, wild animals were not the target of control, but it was the target of a protection, more or less. Under those circumstances, if from the very beginning we had had the notion that wild animals may serve as vectors of diseases, then when we think about the protection, there may be some ideas of how we should control, how we should prevent high density. But we didn't have such a forum to think about these things. That's what I feel. Furthermore, when it comes to the research on biodiversity conservation, well, the term of biodiversity was coined in 1985. Therefore, the researchers of biodiversity are in principle young researchers. Before that, when it comes to ecosystem preservation, it was focused on the rare species conservation or prevention of endangered species to disappear. These are the things we had to think about. For example, keeping them in a zoo outside of the habitat. The protection and the conservation was the focus of the attention. But now that we have COVID-19, as Dr. Goka mentioned, we have come to realize things which we were unaware of. We had beautiful nature left, and we entered into this beautiful ecosystem without any hesitation, and that could be one cause of what is happening now. With the lessons learned, we are trying to identify the causes, and that's what is happening in ecology. An urgent task that we need to involve in, which is still very difficult, is when it comes to ecological destruction or ecosystem destruction. We know it serves as a cause, but the question is what remedies we should have to resolve the situation. We have no clue at the moment. We still even know how to approach this issue, but it is a task we really need to work hard on. And that's all from me, Dr. Goka. Yes, in principle, SFTS by tick is rapidly increasing recently. Before COVID-19, this had been a major topic. With the COVID-19, we have less attention on SFTS, and this new appearance of infectious diseases, we still don't know much about the origin, but the fact that we have these diseases mean that there are more contacts between ticks and humans. That's possible, and it is the wild animal that carries them. As I said in my talk, wild animals are coming close to human society. In case of Japan, there are farmlands and cultivated areas that were abandoned. And with the relaxed management, we have more wild animals entering that area. In the past, we had a zoning, and with the open land of cultivation, prevented the animals, wild animals, to come in to live in harmony. But now that these lands were abandoned, we have wild animals entering, and we also have exotic species like raccoon, and we have more contacts with the ticks. Dr. Okabe's project, I think, aims to identify such a problem, but there's a possibility that this contact may continue in overseas, just like in case of COVID-19. We entered into a nature in the hinterland, so to speak, in Japan, to have more contacts with the wild animals and pathogens. In any case, we have disrupted borders between humans and wild animals, and that may be the basis of emergence of infectious diseases. We need to think of how we draw a line. The concept of living with nature was very vague, even in the Ministry of the Environment. They just talk about the protection of animals, and people think about animals like something adorable, like Winnie the Pooh as an extension. You feed them. In overseas, people go into tropical rainforest, and looking at that nature is reflected as a love toward nature, but in fact such an act destroys the balance and a border between humans and nature. As I said in my talk, zoning is important. It's not that we need to build a fence, but humans' share and animals' share should be set forth. Excessive exploitation should be avoided. We need to have a good control of nature. People have to have a good control of where they live. And as we are surrounded by nature, we need to think about the way we should live. And that, I think, is the starting point. That's what I feel through the issue that we are facing at the moment. Thank you very much. We have heard some challenges and difficulties and what need to do from your own perspective of individual specialities. We have 4 speakers with different disciplines and backgrounds. As I listen to what you have to say, collaboration is important. It's a human disease, but the treatment is not the only thing we need to do. We need to change human behavior. We need to manage wild animals. We need to think about vectors and pathogens, where they are. We need to clarify all these things. Namely, the approach of One Health is extremely important. That's, I think, is what we come to realize. So once again, there may be some duplication, but from the perspective and idea approach of One Health, if there are some specific examples, can you tell us as to how we can incorporate the approach of One Health to control zoonosis in the future? With the keyword of collaboration, I'd like to get some comments from you. Anyone can start. Shall we go through the same sequence? Would that be acceptable? Or if there's anyone who'd like to speak first, perhaps you can raise hand. Yes? Then Dr. Yano. Just like in the current case, the route of transmission should be identified. That, I think, is most important. Once we know the cycle of transmission, we do know where to break off for efficient protection of human health. That could be the starting point. And as Dr. Goka mentioned, we should have zoning. We shouldn't touch wild animals. When it comes to biological conservation, it also means the conservation of pathogens that should be educated. You shouldn't just adore animals. If you touch red fox, you may develop echinococcosis. That education is carried out in Hokkaido, but people from mainland is not aware of this. So the conservation means conservation of pathogens because wild animals carry pathogens. That kind of education also is important. And once a case develops, you should have a good coordination with the health center, which has been a challenge in COVID-19. Related government authorities and bodies need to be collaborating. We need to develop talents who can take a leadership in coordinating these efforts. That's what I came to realize as we see the COVID-19 situation. Thank you very much. We tend to focus on disease control mainly. So when it comes to conservation, I realized that we were paying less attention to conservation. So that I think was a very precious comment. Thank you very much. Anyone else? And then perhaps, yes, Dr. Goka. In principle, the government need to realize that this is a transdisciplinary issue. In short, MHLW took the leadership, but the Ministry of the Environment need to get involved. We have avian flu and swine fever with much damages on Ministry of Agriculture, so they should be involved. When we talk about how humans move, and then METI and Ministry of Foreign Affairs need to get engaged. So the government as a whole have to work together for control. I often say jokingly in my talk, the Shin Godzilla had special headquarters for the control of unknown giant animal. Something similar has to come up with. This virus of COVID-19 is not very scary, it's serious, and it attacks the advanced medicine because we need to protect life. We only have a choice of how we can protect and save life, but during Edo, people died earlier. This may not be a scary virus if we had it in Edo period. Biologically speaking, it was not a scary virus, but the human society was disrupted so much. Then we should be as prepared for a scary virus. Like in case of science fiction, we may get into the worst scenario. So it is important that the government need to consider this as a national security issue. Internationally, it is a matter of sustainability of human society. So it is not just that we should have a narrow scope of a disease that should be treated by doctors. All disciplines need to get involved. It is not just biology, but economy and society. All these perspectives should be incorporated in the policy making. And that, I believe, is the starting point of One Health to move forward. From researchers' point of view, we have people from NIID, from Environmental Research Institute and Forestry Research Institute. It's the first time that we have so many disciplines getting together. We have many silos. When we work with NIID, we have more problems related to contract rather than the contents of the project. So we have sustained silo. This should be really destroyed with COVID-19 because something may occur in the future that is even more serious. It is the policymakers' mission to seriously tackle this, and that I think is the premise of promoting One Health. Thank you very much. including ourselves, under government supervision, we are immersed into this silo structure. And I realize that we need to think about this. Dr. Goka said in his talk, the question of how we should engage humans is, I believe, important. As I listen to these comments, why is it that we cannot break the silo? It may not be a research project, but this is important. And the relationship with economic activities, as Dr. Goka said, what we do with economic activities. When we think about all these matters, as Dr. Yano mentioned, education to start with needs to be reformed to make a step forward. And that's also a new hope that we can find out. Having said that, we still focus on infectious disease, and Dr. Maeda has been working hard on this area, although you may have many challenges and issues. So, Dr. Maeda, it's not my personal view, but a top-ranking infectious disease researcher from China said after COVID-19, it's COVID-20 something. So there may be a series of similar events. Maybe they have already found a similar virus, I don't know. Dr. Goka talked about the infectious disease originating from Japan. Those of us Japanese tend to be clean against infectious diseases, but there are many infectious diseases, but there's a contradiction that they like cleanliness, but not much investigation and survey are underway. In case of rabies that I work on, that's one example. Japanese people are not mindful about rabies. Like in case of echinococcus, overseas it's taken for granted. It's a common sense that you should not get close to animals, but young people are not mindful of this. What I'm saying is that we have pathogens carried by wild animals, but there are many pathogens around us. We should be aware of this fact. We shouldn't be excessively fearful, but we should always keep in mind that there are pathogens around us. We were surprised with the enormity of COVID-19, but we are not too surprised to see this happening. One other point, we all need to once again recognize our situation, and as I said earlier, the fact that the researchers were unable to predict this is a part of criticism, as researchers work hard for prediction. So we need to, for example, use information and artificial intelligence to predict where the next incidents may take place. The paper is available on the prediction using AI. So it could happen any place, but we, mankind, has the wisdom to work on prediction, and we also need to learn from the lessons after the situation over. We need to learn from the lessons. Sorry for focusing on just infectious diseases, but we have to really work hard on infectious diseases. We need to evolve we as researchers, including interdisciplinary collaboration. It's not a good summary, but I just want to say that we have many lessons we have learned from what is happening. Thank you very much. Under the COVID-19 pandemic, it's not that we can come up with a conclusion, but COVID-19 is not the last zoonosis that we face with. There may be further deterioration of biodiversity ecosystem. There may be faster accelerated pace of people and materials moving back and forth. It may be beyond our control for the emergence of infectious diseases, but we need to think about what we can do under those circumstances. It's not that it should be Dr. Maeda only, but all researchers and scientists must think about what we can do. Under the principle of One Health, we need to think about how we deal with nature and ecosystem and how we manage it under the concept of One Health to avoid further infections. I'd like to work on this with all of you. Thank you very much. Thank you very much.